Hi, my name is Karen Brent. Today's date is May the 24th, 2019, and I'm going to do an experiment in regards to my microbiology 225, week 3, in the bacterial morphology and staining technique. And exercise 1 is the direct negative stain. And I have my slide marked with the 1 and direct M, meaning mouth. I'm going to take my sterile swab and I'm going to swab my gum area and teeth. And I'm going to smear the specimen on the slide. And I'm going to let it air dry. And once I let it air dry, I'm going to light my candle. And I'm going to take the slide and I'm going to do a heat fix on the sample with the lighted tea candle. And I'm going to pass the slide over the flames 5 times for no more than a few seconds, and allow the slide to cool for 10 minutes. And then I'm going to add 1 to 2 drops of my crystal violet onto the slide. It's kind of hard because I'm trying to do this by myself. And this is the slide. I'm going to let it air dry. I'm going to light my candle. And like I said, I'm going to pass my slide through about 5 times, for a few seconds each, allowing it to dry and cool for about 10 minutes. And then I'm going to add a crystal violet gram stain to the sample. And once I'm done with that, I'm going to go ahead and place my sample with the staining into my staining tray, which is here. And I'm going to gently rinse the slide with tap water by holding it at an angle over the stain tray and allowing the water to flow over the stain on the slide and into the tray. Doing this until the liquid stain is removed from the slide and the water runs clear. And then I'm going to gently blotch this specimen with a paper towel to remove excess liquids. And by doing that, the drying a slide with the paper towel after the staining. And once I'm done with that, I'm going to move on to my next preparation, which is the 2-M. And I'm going to go ahead and I'm going to take the Congo red and I'm going to put a drop or 2 on this slide here. And I'm going to pretty much, with that, I'm going to use the scissors to cut it. I'm going to do a single drop and then gather a second sterile swab and do my teeth and gums again and place that swab in the Congo solution and use another blank slide to take that solution and drag it down off of the slide to get that slide prepared for microscope. And then I'm going to take my E. coli slide with my E. coli solution here. And I'm going to sterilize. And I'm going to use a sample of my E. coli into the circle. And I'm going to repeat that for each of my broths here. My staph, my yeast, and my E. coli. I'm going to put that back in the alcohol as it's, it's prepared for that particular exercise. And once I'm done, I'm going to do the differential staining with all the different stains as well. And I'm going to go ahead and prepare each of my slides with the different stainings and repeating with the fire to fix the heat to dry. And then I'm going to go ahead and put all my slides on the microscope. And then I'm going to go ahead and take pictures through my lens, optical lens, showing the results of my experiments. And that will be in my lab report. I'm going to add 2 to 3 drops of each stain, waiting 60 minutes, and rinse the slide and return the staining tray. And I'm going to keep repeating those steps until all my slides are prepared for the microscope and pictures. And once again, my name is Karen Brent. I'm in microbiology 225. Today is May the 24th, 2019. Thank you for allowing me to present my lab experiment with you. Thank you and have a good night. Bye-bye.